All right, antibodies, finally, which are derived from what types of cells again? B cells, very good. Now why would a B cell make an antibody? What's the purpose of an antibody? Let's talk about that. It is to connect to antigens. Connect, it's a nice little mnemonic and acronym that'll help you remember what antibodies do. So the C stands for complement activation. We'll finally talk about the complement pathway. It's a pathway that complements the other immune cells. That's why it's named that. And it furthers inflammation, so things like C5a help out with inflammation. Opsonization, that's what the O stands for. Opsonization, that's a big word. What does that mean? Opsonization, I think in Greek it actually means boiled meat. So just like when you boil meat to soften up food and make it easier to chew and consume, antibodies actually make bacteria easier to consume. That's what opsonization is. Here it is. Here's a bacterium. When antibodies bind to it, and specifically IgG, that's an opsonin, it's kind of like sticking a toothpick into a slippery bar of soap. It gives you something to grab onto. It gives the macrophage something to latch onto, because it's trying to grab this slippery, and let's say this bacterium has a capsule. It's all slipping around. It can't get a good grip on it. So it sticks an antibody in there so it can pick it up and eat it. Okay? It's using antibodies like little grappling hooks. So that's what opsonization is. The N for neutralization. So here again we have a bacterium and we have antibodies coming in on all sides, trapping it and preventing that bacterium from attaching and causing problems and invading the tissue. So that's what antibodies do. Okay, let's briefly talk about the structure and function of antibodies. First of all, what is this bond that holds the two halves of an antibody together? It's a disulfide bond. Okay? Just remember that. And in general, there are two main sections of an antibody. There is the FAB portion, and AB stands for antigen binding. So this is the fraction that is antigen binding. And this actually binds to pathogens like bacteria and viruses, and thus it must be variable because it has to bind to every type of bacteria and virus. So this part changes. The FAB part is variable. And then there's another section, and that fraction is called FC, and the C stands for constant. So that's the constant fraction. That part does not change depending on the type of antibody. So every IgG, for example, has the same FC part. Every IgE has the same FC part, but they have different FAB portions, okay? Because they have to be able to bind a bunch of different types of antigens. Things to know about the FC section is actually the rule of C's. So it's constant, like we just said. It possesses the carboxy terminal end of the protein chain. It activates complement that we have yet to discuss. And it has little carbohydrate side chains branching off of it. Okay, so remember all the C's for the FC. Okay, so let's continue talking about antibodies, and now let's talk about how they're made within B cells. So remember back when we talked about VDJ recombination? When we said that innate versus adaptive immunity, that with adaptive immunity, we had a specific response because the immune cells had time to develop a specific antibody that reacted against whatever pathogen was attacking the body. So, if you just think for a second, I mean, there's got to be millions, potentially billions of different types of pathogens out there. And your body has to be ready to handle all of those billions of types. How does it do that? Well, it does that through permutations and randomly reassorting the genes that make up and eventually synthesize the variable parts of antibodies. So, for example, here's a heavy chain. There's a, V, there's a variable part, there's a D, stands for diverse, J is the junctional part, and C is the constant. The constant part doesn't change much, but the V, the D, and the J sections all swap around and are spliced out and joined together randomly so that there are essentially infinite permutations that can be developed from a number of genes within B cells. So this allows B cells to make a unique type of antibody. So first of all, the heavy chain is synthesized, RNA polymerase transcribing, and eventually synthesizing the heavy chain. And this also occurs for the light chain as well, except instead of VDJ, it's just VJ recombination. So again, The genes are spliced out and randomly joined together. And you can just imagine all the numerous possibilities of combinations of light chains and heavy chains and combinations of B's and D's and J's that can develop essentially an infinite number of antibodies. And this allows us to bind any type of pathogen because there's a good chance that it just takes a while for it to find it, but there's a good chance one of your B cells is going to bind to whatever pathogen enters your body. Just like I said, it takes a little bit for it to locate it, and once it locates it, then it has to get stimulated. Remember how we talked about before, it's stimulated by the T cells in cytokines and co-stimulation, and that will cause this B cell to increase in number and say, hey, this B cell is making the right antibody. We want this guy to keep making more copies of himself because he has the right antigen that's going to stop the pathogen from killing our body. Make sense? Pretty cool, actually, I think. So there's the light chain combining with the heavy chain, and voila, have an antibody. It's pretty neat, I think. Not to boast about my cool animations, but I think it's pretty cool. So, again, what antibodies are on the surface of B cells? MDs are mature, right? IgM and IgD. So MDs are mature, that's how you remember that. Most are mature. I might be an exception. And then, of course, a B cell is an antigen-presenting cell, so it bears the MHC2 complex as well. Alright, so now let's discuss the different types of antibodies. So like when I say IgG or IgE, those are both different isotypes. IgG is an isotype of antibody. And we'll actually start off by talking about IgG, and we'll go down in order of prevalence. So we'll start with G and end with E. And that's how they go. And from greatest amount to least amount in your body goes GAMD. Can I remember that? And for each one, like for IgG, I'll try to use the letter of the antibody to try to help you remember certain characteristics, okay? So, for example, for IgG, there's the greatest amount in your body. There's a growing response. And by this, I mean, what is the initial antibody that's synthesized from B cells? IgM, right? Then later on, after there's class switching, IgG is most prevalent. So that's what I mean by growing. It grows later, after the immediate antibody. And finally, it's given to infants through passive immunity. That means that the infants actually don't synthesize the antibody themselves. They're given it through the placenta from their mom or by doctors in the hospital if they need it. All right, next one, IgA. A, you can think of, prevents adherence, with a capital A, to mucosal surfaces. And you add two antibodies together because that's what it is. It's a dimer of two antibodies, all right? And here we go. Here's a mucosal surface. That's where you think of IgA. So let's say we're dealing with your intestines, and we have a plasma cell that makes antibodies, of course, and it makes some IgA. But before this IgA leaves into the lumen of the intestine, it has to acquire this secretory component. And for some reason, step one thinks that this is important, so know that a secretory component is involved in secretion of IgA. All right, so it's bound to that secretory component that kind of looks like an S too. And then it can go and bind to pathogens and prevent them from adhering. IgA adhere. Good. Next up, IgM. Remember, it's the immediate response. The first antibody made by B cells. It's macro, mega, it's huge. Remember, it's a pentamer. 5 antibodies strung together. And so you don't want to get in the way of that sucker. So typically what IgM does is it's the first responder. It sort of traps pathogens and keeps them hanging around until B cells can start making and class switching into IgG. All right? So they trap pathogens. All right, next one, IgD. Remember, MDs are mature. It's on the surface of mature B cells. But besides that, don't worry about it. IgE. Think of allergy, okay? Because, as we already discussed before, B cells that are stimulated by what type of T cell again? Remember? T helper type? TH? 2. Achoo, right? Good. And what interleukin's involved? Interleukin 4. Good. So before we even talk about that, you know what's coming. So B cells start off by making IgM, but then these T helper type 2 cells come along and release their interleukin. Sure enough, interleukin 4 and possibly 5 go and stimulate the class switching of this B cell to start making IgE now. And again, stick these on the surface of mast cells, cross-linking, degranulation, allergic response, histamine, vasodilation, eosinophils are attracted over there. By the way, what interleukin attracts eosinophils? Interleukin 5. Okay, we'll talk about that later. All right, so easy flash quiz. What are the different types of antibodies? And in order of frequency, IgG, A, M, D, and E. GAMD. Very good. So now let's briefly discuss antigen type. And depending on the type of antigen, there will actually be a different type of memory response to that antigen. I'll show you what I mean. So there's either thymus dependent or thymus independent. And remember, with thymus, what are we really saying? Thymus means T cells. So T cell dependent or T cell independent. And if you involve T cells, you get a more robust memory response. So we want thymus dependent for better memory. And here's what we mean. So here we have a cell, just some type of cell, that is infected with a virus. Again, we already discussed how it degrades the virus and sticks a piece of it onto its MHC1 receptor, right? What cell will come along? What's this? It's either a natural killer cell, but we're talking about T cells here. This is probably a CD8 T cell then, right? Right. All right, so then cytokines are released, and we have memory. And this is because of the antigen type, actually. So this is a proteasome, right? This was a proteasome. And proteasome implies that it degrades protein. So this antigen needs to be a protein. It needs to be amino acids for you to get memory. Otherwise, you can't degrade it like we see here in thymus-independent antigens. So let's say that there's this bacterium and it has just a bunch of polysaccharides that are not made of proteins. And a piece of them is ingested by this cell, and it can't be degraded properly. So we don't have as robust of a memory. We need this T cell interaction to have a robust memory. We need these cytokines to be released to have that memory.